Sveiki visi susirinkusieji, iš tikrųjų labai smagu matyti gausią auditoriją. Labai tikimės, kad šiandien mums pavyks sėkmingai pasidalinti įdomiais pranešimais, idėjomis, įvairiais atradimais. Tegul ši diena iš tikrųjų būna ir šventiška, ne tik tai darbinė. Ir tikimės, kad iš tikrųjų šita diena jums paliks įspūdį. Welcome to the postdoctoral conference at our center. We are delighted to be together today and to share various ideas and discoveries. And we hope that each of you will find something valuable and inspiring at this conference. And now I want to invite our director to take some words for the participants and for our guests. Please.
Labas rytas visiems. Good morning, everyone, dear guests, friends, and colleagues. Good morning and very warm welcome to Nature Research Center. And I'd also like to extend a special greeting to those joining us virtually on the other side of the screens. And it's a great honor and excitement for me to open this postdoctoral conference. And I would say now it's in third edition, because it's 3 years we have this conference. And when we organized this first conference 3 years ago, we envisioned it as more than just an event. We hoped it would become a tradition, a key platform where postdoctoral researchers could showcase their groundbreaking work and build bridges across disciplines. Today, I am proud to say we are fulfilling this vision. As I look around and this gathering, and when I'm looking at the program, I am in awe of the diversity of research being conducted at our center. This year's theme, Discoveries in Cultural Landscapes, Climate, Biodiversity, and Evolution, reflects the vast array of questions our researchers are exploring, from the agricultural landscapes of the Roman period to Holocene climate reconstructions, from studies of freshwater biology to the evolutionary history of crustaceans, from blood parasites of rodents and birds to biodegradable nanosolvents and probiotics. Our center is a hub of cutting-edge science. We have scholars joining us from different countries, all bringing their unique insights and expertise, making this a truly international and multidisciplinary event. The Nature Research Center stands as a testament to our shared commitment to advancing science and expanding the boundaries of knowledge. Over the years, many discoveries have been made within our labs, and as we convene today, we continue this proud tradition. This conference is more than just an exchange of data. It's a space where new ideas are born and where collaborations emerge, which will push the frontiers of research even further. Throughout the conference, we will hear from our speakers, each sharing insights from their own research journey. I encourage everyone to make the most of the presentations. Please ask questions, engage in discussions, and let your curiosity guide you. This is an incredible opportunity, not just to learn, but also to network, to build friendships, and to forge professional relationships that will last long after this event concludes. And finally, I want to express my deepest thanks to the organizing committee, whose hard work and dedication have made this conference possible. And as we begin, I hope this event will spark innovation, collaboration, and discovery for all of us. May the ideas exchanged here today lead us to the new breakthroughs, and may the relationships we build help shape the future of our research, both here at the center and beyond our boundaries. And thank you all, and let's get started our conference. So thanks.
Yes, the broader discussions to be continued after the conference in the adjusted room with a cup of coffee. And I want to ask, want to say that for every presentations and for questions, you have 20 minutes. And let's try to stay within the time limits. And let's start our conference. And the first presentation, please. The first presentation is the year of Karolis Minkevičius Agricultural Landscapes of the Roman Period, an update framework, Supervisor Dalia Kiselienė.
So, good morning, everyone, and we'll start today's conference with a small talk about archaeology and what geosciences can contribute to this field. So, we'll talk about the explorations of the agricultural landscapes during the Roman period in Lithuania, and just a very brief recap. Archaeologically, Roman period in this part of the world dates from the 1st to 400 Cal AD, and this is very interesting period because a lot of things are happening at the same time very rapidly. So apart from the influence from the Roman Empire, we are seeing changes in settlement patterns, transformations in the social environment, changes in the landscape, and so on and so forth. And what's really interesting is that we're seeing some of the most significant changes happening in agriculture, primarily the introduction of new species, so oat and rye, also a very rapid increasing rates of deforestation across the landscape, and a very sharp decline in the previously cultivated crops, primarily common millet. But the problem is that most of this information comes from the archaeobotanical record, so pollen and plant macrofossils. And this causes a bit of a problem because the evidence is quite fragmented. It's not very evenly distributed, and this causes all sorts of distortions. And we can also see this from the sites we have explored, that there is a huge array of diversity going on. Some of these could be resulting from the human actions, so different pathways of the deposition of archaeological record, whereas some of them could be explained by the local topography, but it's really difficult to get a sort of justified sense of what's going on across the region. So in order to tackle that, we decided to look at the chemical composition of the crops discovered in archaeological record themselves. And we did that by looking at the two different stable isotopes, carbon and nitrogen, because these could give you direct information of the environment and cultivation technologies used for growing crops. So carbon, well, all plants absorb carbon by the process of photosynthesis, and different types of isotopes are absorbed. Carbon-12 and carbon-13 are one of them. And during the photosynthesis, plants tend to discriminate towards the lighter isotope. But the thing is that if the rate of photosynthesis is inhibited for some reason, the values of carbon-13 tend to be elevated. So this can give you an insights into irrigation, so water availability, and also sunlight availability during the vegetation process. And nitrogen, on the other hand, is more linked to the human action, so primarily manuring, because during the decomposition processes, which results from the application of manure to the soil to increase soil fertility, we discover the elevated nitrogen values, whereas if the land is fertilized using the ash resulting from the burning down of the forest, these values would not be elevated. So we started looking at these isotopic values, trying to see whether there are any differences compared to the earlier periods. And what was really helpful is that this is an ongoing project, and we have already gathered some data from the first millennium BC, and we started by looking at one taxon in particular, which is barley, primarily because this is the main cereal crop throughout the prehistory of Lithuania. And again, this is the only species that was present in all of the analyzed contexts. And the huge letdown was that we don't see any differences in either nitrogen or carbon values, because apparently most of the prominent agricultural technologies were already implemented prior to the start of the Roman period. That said, we did find something very curious and probably much more interesting, which you can see right here, circled in green and in red. And this marks the disparity between two different crop species. So green is barley and the red is rye. And what's really interesting is that in some of the analyzed sites, we can see that nitrogen values showing the soil fertility are much lower in the case of rye. And again, this pattern is repeated if we look at the water and sunlight availability. And this very clearly shows that these two species at the same sites are cultivated in very different landscape setting. So rye being grown on much poorer soil with much less access to water and sunlight. And if these two species were grown in a similar landscape, you would expect to see these two being plotted much more closely, and especially if you factor in the anatomical differences between barley and rye, with rye being much more prone to accessing nutrients from the soil in more harsh environments. Another interesting, and this really feeds into the archaeological data, which sort of already indicates that most of the farmsteads during the Roman period are located in the river valleys with the sandy, light soils, which are constantly fertilized by the alluviation processes, because we already have discovered traces of arable land from this period in the river valleys. But what's really curious is that this pattern is not uniform across Lithuania, because we also have sites where all of the isotopic values of different species are plotted closer together. So we know that at least in some cases, all of the crops are grown in pretty much the same area. Whether this is because the amount of fertile land is abundant, or there just isn't enough of the demographic pressure to expand into a more difficult landscapes, is still unknown, but we see that there is a difference between these two. Again, we dated all of the analyzed contexts, trying to see whether there is any sort of linear progression as we move into later periods. We managed to group sites into 3 different time frames. So start of the Roman period, the end of the Roman period, and the start of the migration period, which is more along the lines of the mid-1st millennium AD. But the thing is that the sites are all over the place and there is no linear progression to be found. And probably the last and most curious observation is that in the archaeological record, we also see the failed attempts at trying to adapt to the landscape, different landscape settings. This is one example from central Lithuania, a site called Skudani, just south from Elektrėnai. And this clearly shows that all of the crops are grown in the same area. So barley and rye and all of the species. The nitrogen values indicates that this was a somewhat fertile landscape with also a lot of human input. So it would seem that this was a perfect place to stay, but what's really curious is that this is a very short-lived settlement, which spanned just around 50 to 75 years. And it was abandoned without any apparent reason. So no signs of fire, no signs of assault. What's really interesting is that people never turned to this landscape. So one possibility could be is that since the landscape has many small hills and the topsoil is very clay rich, hence it's poor draining. This causes a lot of problems trying to engage in a cultivation of for winter cereals, so primarily rye. And looking at the composition of the Acer botanical remains at the site, we also see that while rye is present at the site, it's only cultivated at very low quantities. Another example comes from Western Lithuania site called Melanie, and this offers a very interesting comparison between the Roman Iron Age horizon at the site, so the very first stages of human inhabitation, compared to the Viking period, which is 1,000 years later. And if we look at the isotopic values on the left-hand side, we see that during the Roman period, either there were no attempts of manuring of the local soil, or the site was abandoned before these efforts could kick in. Because at this site, as compared to Skudani, we see signs of sudden abandonment caused either by an accidental fire or an assault. And if we compare the values on the right-hand side, which are from the same site from the later period, we already see that there are traces of human maintenance of the landscape. And again, we see the very same pattern being repeated with barley having an elevated values in carbon and nitrogen, as opposed to rye, which is very down at the bottom. So one of the main takeaways is that when we look at the agriculture throughout the Roman period, we can't really talk about the single uniform agricultural landscape. We should really focus on small, different regional landscapes, but all of these have some common denominators. So we're talking about the permanent field agriculture taking place primarily near the settlements on an open, well-irrigated landscape with constant maintenance, including manuring, tillage, and so on and so forth. And some of the crops in some of the sites being cultivated somewhere elsewhere. The most likely example would be the cultivation of rye in Sweden, which are in a more distant areas and which would not require a constant input. And I would suggest that this probably all reflects the attempts of local populations to adapt to either to both the different local geographic settings and also very challenging conditions when it comes to agriculture in this region as a whole, because as we know, we have very hot summers, freezing winters, and lots of rainfall. But what we also see is that sometimes these efforts had drastically different results, leading to different rates of success. So thank you very much for listening. And now I would be very happy to answer any and all of your questions.
Jūsų tas taškas, ar čia yra vienas matavimas, ar čia apibendrinta statistiškai?
Ne, čia yra vidurkis su standartiniais nuokrypiais.
Ir iš kiek panašiai taškų?
Kiekvienos vietos, kiekvieno konteksto nuo 5 iki 10 matavimų kiekvienai rūšiai, bet siekiant tarp 5 ir 10 matavimų, bet problema yra ta, kad išlikimas skirtingose vietovėse yra labai skirtingas. Ir tokiose, kaip ten Kernavė, jų galima pridaryti daugybę, o kai kuriuose apie 5 ar iki 5. Yeah, in Center for Physical and Technical Sciences in Saulėtekis.
Ačiū už pranešimą. So you showed very nice pictures of landscapes. And I expect that this is reconstructions. And how these reconstructions might be close to life that time. Do you have some expectations about this? Or this is just nice pictures and that's it?
I mean, the problem is, again, that the archaeological record is quite scarce, so we can only rely on some of the sites which are, well, most excavated. The primary one is, again, Kernavė. So looking at Kernavė, we're pretty sure that this is how the landscape looks like. So the more or less open valley, the settlements are located in the valley, the fields are next to them. And we try to rely on this example, trying to understand all of the fragmented ones.
So is it a scientific background for that? Or this is your expectation?
Yeah, looking at it all derives from two bases. One of them is the archaeological one. Another is the more environmental one. So the environmental one is based on the analysis of plant macrofossils, so seeds and fruits. And again, this poses some problems, but there is some basis for it. And the archaeological side is a bit more well understood because just the amount of material is abundant.
And another question. So the work has historical significance. We need to know how our ancestors lived. How your study might be related to current days?
I guess one of the main takeaways is that people nowadays, as in the past, should be very, very careful in the ways they manage their landscapes, because we see time and time again in archaeological record when people settled in different areas start changing the landscape to fit their needs better for the defensive purposes and from agriculture. And we often see how this all leads in a very rapid deterioration of the landscape, leading resulting in the need for the local population to abandon the place. So we have a very nice example, which is from an earlier period, from Western Lithuania, where everything was destroyed in several decades. Kernavė is another example where the changing of the landscape on the upper terrace led to a rapid erosion of the topsoil, and again led to the abandonment of the landscape. So this is probably one of the main things that we should learn from the historical lessons.
Thank you very much for the introduction. I would like to point out that my review chat after a whole year of study. So this is a document approach, so I will have the results. Yeah. So during the presentation, I will briefly explain the motivation which is behind the project. Then I will come to why this is study administration, why this is why we doing it for it. And I will also speak something about. Do you hear me without microphone? I need to do the okay.
So, yeah. So I will briefly describe the methods which I am going to use for dating, or which I am currently using. And finally, there will be a few slides about the results. So why it is important to study past glaciations? We can say that it's obvious in the times of climate change that it's important to understand the interactions of atmosphere, cryosphere, and hydrosphere. And we have some paleoclimatic records, which are mostly like this stack of oxygen isotopes based on ice cores or cores of marine sediments. These are globally averaged, and these do not mirror specific changes in specific locations, like specific ice sheets. And we have a very scarce record from continents, which is because these glaciers are working like bulldozers, and they're eroding and recycling previous sediments. So these records are quite scarce, and we have some estimate for Lithuania, which is between 0.9 and 0.7 million years. But this is, again, mostly based on marine sediments. So the motivation of this project is to provide some first radiometric dating of the onset of glaciation in the area of Lithuania. And so what is Lithuania? Lithuania is very specific because it consists of up to 300 meters thick quaternary sediments, what is quite unusual. So you can see on the figure on the left side alteration of glacial and interglacial layers, which are stuck one above another. And you can also see the lowermost yellow horizon, which is considered like the fluvial oldest pre-glacial accumulation, and then the light blue one, which is the first moraine, like the first preserved glacial sediment. These sediments were previously dated by magnetostratigraphy, but without any radiometric dating. This is also a methodological problem because it's really complicated to get some radiometric age for these sediments. And the magnetostratigraphic dating and previous research was done on these outcrops, which are located near Anykščiai city in the eastern Lithuania. And these outcrops are also included in my study. So you can see some logs here. And as one example, you can see a Šlavė outcrop. And more specifically, I'm focusing on Daumantai, Vetigala, and Šlavė. And these outcrops were studied before more than 10 years. So you can see they are quite overgrown and they require quite a lot of excavations in order to be able to observe some beds. And apart from these outcrops, which are, let's say, natural, I'm also focusing on Anykščiai quarry, which is continuously excavated because of the sands. So this is another example of Vetigala outcrop. And these outcrops are quite big, quite extensive. So I'm using a drone photogrammetry to get some 3D models of the outcrop. This helps me to correlate different parts of the outcrop quite precisely and also to observe the changes in depositional record in a very detail. And I must say that I get this PixPro software for this for free from one Lithuanian developer of the softwares. Another example is the Anykščiai quarry, which is much bigger, and you can see it here. And the lower level exposing these pre-glacial fluvial sands, which are white, and a level above exposes moraines, which are the first glacial, which is preserved. And you can see here output of another software, which I'm using for 3D interpretation of depositional record. And you can also see a place of one of my profiles, which is on the left side. Interestingly, you can see in the center flat plane horizon, which is very important for my dating because it consists clays, apart from other parts which are mostly sandy. For detailed study of facies, I'm doing these lacquer peels. The lacquer peels are preparates made from the wall of the outcrop by applying this bison kit glue, which is originally used for preparation of shoes. And this allows me to, from one side, to preserve the positional record because, for example, Anykščiai quarry is continuously excavated. So something what I am studying now will be lost in a few months. So in this way, I can have piece of this depositional record in my office. And another good thing on this is that I can study the structures much more in detail. Here you can see one example on the left side, where you can see cross-stratified sands, and these sands originally form like a subaquatic dune, which is migrating on the bottom of a river. And these dunes or cross-stratifications are very important for me because like you can see on the 3D model, we are able to measure their orientation in depositional record. And in this way, we are able to reconstruct the direction of paleo flow. And in this way, to reconstruct the properties of a paleo river network. The outcrops which I am studying are quite far from each other. It's a few kilometers in between. So in order to achieve a detailed correlation, I plan to use construction of, let's say, 3D maps and geological profiles through archival boreholes. I acquired more than 900 of these lithological logs of boreholes from the Lithuanian Geological Survey by the help of Aldona. And I have just one small problem that all these descriptions are in Russian. So I'm too young to speak Russian. And I'm now trying to find automatic solution to translate these descriptions because it's like hundreds of descriptions. And obviously I can use Google Translate instant camera translation, but it's quite inefficient for a single page. So this is something I'm working on now. And okay, what about the dating methods which I'm using? These are based on cosmogenic nuclides. So we have a constant flux of cosmic rays, which are interacting with matter in atmosphere and on the Earth's surface. And these cause nuclear reactions and produce cosmogenic nuclides. There is a number of cosmogenic nuclides, but I am using beryllium-10 and aluminum-26, which are produced in quartz, which is exposed on the surface of the Earth. And also I'm using beryllium-10, which is produced in the atmosphere in the same way, let's say, like radiocarbon. And from the atmosphere, it is transported by precipitation to Earth's surface. It's important to say that cosmic rays are penetrative, so they do not stop on the surface, but they are also going into the rock, and the intensity of cosmic rays is decreasing exponentially with the depth. This is important for the first approach of dating, which I'm using. It's based on depth profile exposure dating, because we are taking samples in regular intervals with the depth, and we are trying to capture the exponential decrease and model it. And the model, or let's say the shape of the curve, is proportional to the age of exposure of some sedimentary unit. If we have several depositional units which do have different age, we also have several exponential decreases one above each other. And there is also a problem that we might have very variable inherited concentrations of cosmogenic nuclides. And in this case, we would have scenario which is to the right, a scattered distribution, which is very problematic to model. And this approach is possible to combine with the same samples, same data, and same measured concentrations with the Boreal dating, which is maybe more familiar to you. And this is based on assumption that if you have a quartz pebble exposed on the surface, the production of beryllium-10 and aluminum-26 will be of the same ratio, like a stable ratio of 1 to 6.75. But when you will this pebble have transported by a river, let's say to a cave, or it will be buried by a thick sedimentary succession, the production will be shielded, or cosmic rays will be shielded, and production will be stopped. And the ratio will change with time because these two nuclei do have different half-life. So the decay will change the ratio, and we can use this ratio for dating. So these are the methods which I'm using with quartz samples from sand or from gravel. And completely different method is based on the beryllium-10, which is produced in the atmosphere. It's transported by precipitation to the hydrosphere, and in the hydrosphere it meets with beryllium-9. Beryllium-9 is derived from a chemical weathering of rocks and also transported, let's say, by streams, by rivers. And in oxic environment, these two isotopes are incorporated in oxyhydroxides, which form autogenic phase. This is a very small rim around sedimentary particles. And for the dating, we are dissolving just this reddish rim, which you can see in the picture in the middle. And when the particle is deposited and buried below sediment, the ratio of these two isotopes is changing just with the half-life of beryllium-10. I have to say that for this method, we have to find out the initial ratio from which this isotopic ratio of beryllium-10 and beryllium-9 is changing. And this is done by analyzing, let's say, equivalence, sedimentary equivalence of the environment which we are dating. Just an example, if we are dating river sediments, which are expected to be, let's say, 2 million years old, we have to find Holocene or very young river sediments from the same area, and we are more or less comparing the isotopic ratios between these sets of samples. So together I accumulated quite a lot of samples. Some of them are under processing, some of them are waiting for measurements. I took 29 samples for autogenic beryllium-10 and beryllium-9 dating from outcrops, and 36 for exposure and Boreal dating. And I also took some samples from boreholes, and I didn't mention these boreholes before because I will speak about them on the next slides, and I have first results from these. So what about the first results? I also sampled a few boreholes which are located to the east from Vilnius. And this area is different from the previous one with outcrops because the area with outcrops exposes these sediments of interest almost nearby the surface. Here, the same beds are below 200 or 300 meters thick cover. So I took samples which are marked by stars on the profile, and the yellow ones are already processed. And it's important to say that I also took some samples for the initial ratio, which is mentioned. And these are from Holocene Schwentoy River, which is flowing nearby the area of the outcrops, and also from one section of last interglacial Paleo Lake and from the Nemunas River Delta. So here are the first results, and I can say that they are not so nice as I would expect, but okay, let's say these settings are very complicated for dating because of the glacier recyclation and another complicated boreal exposure histories. So what we can say, for initial ratio samples, we need a very narrow distribution. And these samples are really scattered, what is quite a problem. There is some peak in the middle, but there is just a few samples in this. So I took much more samples from the Schwentoy River, floodplain from different sites. And I hope that I will get, let's say, more narrow distribution, because with this wide distribution, I'm not able to calculate initial ratio and use it for dating. Another problem is that in the middle, you can see samples taken from the youngest pre-glacial sediments, so this fluvial horizon, which is just below the first moraine, and they are of the same or even higher concentrations or ratios comparing to the initial ratio samples. So they are apparently younger than the recent sediments. So what is nonsense? So something is not working in the system, and input of isotopes was violated in some way. So it was not the same in the past like now. But this is different for the oldest interglacial, which is on the right side. In this case, we can see much less variable distribution with some peak. And if we would use the peak of the initial ratio samples and the oldest interglacial, we will get 800,000 years of age, what is quite reasonable. But this is very, very preliminary because it's based just on a very few samples. But we are going to get more values, so hopefully it will be much more precise. So thank you very much for your attention. I just would like to say thanks to everybody who is helping me with the research, to my supervisor Albertas, to Vaida, Aldona from Geological Survey, and also to guys Andrius and Alexei who helped me with the excavations in the field.
The biologist, and I'm interested in these glaciations and their effect on distribution of organisms and so on. So according to you, you would say that around 800,000 years is when the first glaciers were already present, more or less.
This would be more my guess, also without these results, because it's the middle Pleistocene transition, where we have changed from, let's say, 40,000 climatic cycles to 100,000 climatic cycles, which are much more colder. So, and there are some datings also from the European Alps, which are indicating that this was the onset of first real glaciation there. So, but these estimates are really variant. So,
okay,
but these would be my guess.
Okay, yeah. And is it so like before this time there were like no glaciers, or at least like in Scandinavia or in the Alps? Is it?
I can't speak about Scandinavia. I would say that this is very difficult to. Yeah, from a marine record, there is something about Scandinavia, and it indicates that there might be glaciers before, of course, yes. But they don't have any continental record like this because the accumulation is much, let's say, the accumulation space there is almost nothing. So they don't have preserved sediments from this time on the continents.
Okay, thanks.
The next presentation, Dr. Bennett, of the climate reconstruction,
Good morning. My name is Nick Schafstall. Bennett is my middle name. And I'm working on the project Early Holocene Climate Reconstructions Coleoptera from the Eastern Baltic region. I have to mention that this is an individual postdoc project funded by the Lithuanian Research Council. But instead of visiting a lot of different institutes, we did fieldwork with the money. I also don't have a lot of results because I started fairly late. I started this April. And we just finished our field works in May to August. I'm still very busy with the lab work. So maybe next year, at the end of next year, when we have the conference again, I will be able to show you the results. But for this project, we went to Estonia, Latvia, and Lithuania, where we found early Holocene deposits, and we dug up beetle remains. But first, I want to tell you what I'm actually doing and why I'm doing it. The history of Quaternary entomology is quite long, and what it involves is Quaternary. So Quaternary is the last 2.5 million years, where you have the period of these ice ages that Michal also mentioned before. So you have a lot of cold and warm periods, and it is also the time period in which humans started to evolve. So a lot of people are interested in this, and when they were excavating Quaternary deposits, they found a lot of insect remains in these deposits as well. It started early 20th century, mostly in the UK and in North America, a little bit in Sweden, but where you have all these nice layers of accessible glacial and interglacial layers. And these insects that they thought, okay, this is all extinct. They called it Hellephorus extinctus or glacialis and so on. And it took a few decades before people started to realize that these insect remains from hundreds of thousands of years ago are actually extinct, not extinct species, but extant species. They started to compare it more with, like here, with the extant species, and by looking at how maybe these fossil remains of insects change through time, the taphonomy process after death, they discovered that practically all these species are still extant. So they started to work with it, like what does it mean that you have all this continuity of species later on? So they started to work with it, and in the UK they really picked up on this around the 1970s, where because there were some species that they could never find, but eventually they found species that were in glacial deposits living now in the Himalaya. When they worked more on this, they found that there were every glacials and interglacial cycles, there were species assemblages always of the same species that were then all the time together, but nowadays might be living in the Himalaya or might be living in some other mountain area. So the theory was born that beetle species do not evolve. They do not evolve climatically, but they migrate through time. They just migrate wherever their climatic niche is suitable. If they're more cold adapted species, they will move to the Himalaya if it's becoming warmer, and they might come back. If it's colder period again, they might come back again to the same site or to another site, which then suits them. When they found out this, and they found just a continuity, they found almost no exceptions to this rule. Beetle species migrate like many other insect clades, and they just migrate wherever the climate is best. In 1986, North American researchers developed the mutual climatic range method for beetles, where they took the climatic range for all the extant species of North American beetles, which are sensitive to climate. They looked where it is common now. For instance, here there is, to the left, you see the distribution where species A is found in North America, to the right where species B is found. From the weather stations, they reconstructed what is the maximum summer temperature, the minimum winter temperature. So you get climatic envelopes for these two species just below. And if you overlap them, if you find, then you know, if you find these species together, even in a fossil assemblage, assuming that really their climatic niche has stayed the same. You can see what was the climatic range during the time of the position of these two species together. This was quite a popular field in the 1980s and 1990s, where they did reconstructions for how the climate changed after the last ice age, how rapidly the climate changed compared to plants, because plants have to migrate while beetles migrate within a couple of years. So they did great publications on that. But lately, it's mostly been used beetle assemblages for nature conservation. Because apart from climatic niches, beetles have just in general, some species have very specific ecological niches. They only occur in forests, they only occur in some species of trees, they only occur in dung. And some colleagues have tried to reconstruct through time what was the percentage of forest in the UK, what was the percentage of dung beetles in the UK, and try to reconstruct how the landscape on large scales changed through time. So that's in brief the history of quaternary entomology and some of the applications. That's why beetles are a good proxy, because they preserve really well. As you've seen from the fossils, these fossils were hundreds of thousands of years old. If there's no oxygen involved, they preserve that well that you can put it next to a museum specimen and you can say, what is the species? They also provided defined source areas. Some previous experiments shows that if you dig a hole at the site and you find beetle fossils, these species occurred around 500 meters from this site. So you get a very nice local signal of what happened at this site through time if you get multiple samples. On top of that, a lot of species have a very specific ecological and temperature niche. They form a very good paleo-ecological proxy to reconstruct these things. And they can give important supporting information to pollen records, which have a more regional signal, maybe fires. If you add beetles to the sun, you can get a lot of important supporting information. So this mutual climatic range method has mostly been used in Western Europe and North America. This is a picture of a database of fossil beetle records. And you have to imagine that this is the sites, it's just a pinpoint. These dots would mean 50 to 100 kilometers. So if you really think, well, how many sites there are, also taking into account that these are also a lot of archaeological sites, not only paleo-ecological sites. Everything is concentrated in the UK and Western Europe, maybe in a little bit of North America, southern Sweden. And for me, it was really a pity that after the 1990s, no more research has been done. It's on this climatic things. Oh, we found out how it works. Let's move on to something new. But it has never been tested in Eastern Europe. It has never been tested if it can also be used in mountain areas to see if nowadays we got a lot of important studies going on about climate change in mountain areas. Maybe it can be used there. So these more subtle temperature changes compared to glacial interglacial changes. Can the mutual climatic range also be used to that? But no, research has been done. So I took the opportunity to use Eastern Europe as a case study to test it. What is possible, what is not possible if you try it in a different region, different species, maybe early Holocene instead of late glacial. So far during our field work, we went to a couple of sites. We went to Estonia, to a peat mire and to a peat outcrop. In Latvia, we went to a peat cutting site, hoping that the sediment would be also early Holocene. So about between 10,000, 8,000 years old. Also nice, we took early Holocene in my research project to try to avoid any human interference in the beetle species assemblage there as well. In Lithuania, we went to Vente, Pameke and Ula. In the south of Lithuania, there are in the river valley some really nice sites of early Holocene that you can reach. There's not so much, normally there's meters of sand on top of it, but in these river valleys, you can reach this early Holocene outcrops. And in Poland, we are working together to also look at a peat bog there. So these are some pictures, because at least I can show you some pictures of the Estonian sites. This is the, to the left here is the marsh, the peat outcrop and the cut peat bog. The sites in Lithuania, Vente, it's a very small band of organic material in the dunes, but it was dated before. So we know, at least in this Lithuanian sites, how old it was. Pameke, nice layer of lake sediment, and Ula, even a thicker layer of lake sediment, which we took with help of Migle, Andreus, Vaida, local researchers. So then what we're doing is we're going to radiocarbon date these layers again. It's young enough to radiocarbon date. To get the insect remains out of it, I have to sieve it, float it, extract the insect remains from there. Later on, I have to identify the insect remains. And finally, if I can identify these insect remains to species, sometimes genus is enough to know the genus level. I can reconstruct ecological and hopefully climatic niches based on these species assemblages. So this is just a picture. This is a core that we took from Latvia. But you get to this nice sample. A lot of hard work is involved, where you have to sieve, you float it with paraffin oil so that the insect remains start floating on the surface. That is the way to extract the insect remains. Then when you have the sediment, it might hopefully contain a lot of insect remains. I'm still busy on that part. And when you start to sort them, some insects, some can readily be recognized to the species. You find out what the species is, and then you have the ecological and climatic properties of that species, combine it to all the species that you can identify, and then you get a reconstruction, which is the next picture. This is done previously, and what at least to a certain extent can be done from the sites that we excavated so far. Landscape reconstructions will look like this, where you have the number of aquatic beetles, the number of marsh beetles, but normally you also find a lot of forest beetles in this site because it's 500 meters around the site, so you find everything that lived around the site where you're excavating. If the beetles are, if there are some species that are also temperature sensitive, you can look at what is the climatic range of these species as well. And then you combine it and you get the climatic overlap. This is what we're trying to do on Botany sites. It's a pilot, but it will be a great base for possibly future work. I'm going to submit just a manuscript on what we find in Lithuania. And what I will find, I will use to argue what is possible with the mutual climatic range methods, and what are the steps to make further. But we collected so much material that probably many more articles can follow from this. And that's my presentation. Thank you very much. I'm open for questions.
Thank you for your presentations. Prašau klausimų.
Nick, thanks for the nice presentation and good overview. I'm just curious, do you standardize sampling at each one of these sites? So for instance, are you digging out maybe like a 1 square meter plot at each site, and then you're comparing, in that way you could compare the abundance of these beetles per site in a more standardized way?
We try to, but definitely we measure the volume of the sample that we take home. We measure the volume before we start processing it further.
Okay.
So yeah, it's also a thing. You see how many individuals per, let's say, liter of sediment you have in the end.
Okay. Second question: are these insects preserved well enough to do some genetic methods to identify them? Because I heard you mention paraffin oil, and I don't know if that maybe affects the insects that you use. So you use this paraffin oil to make them float.
I'm not sure if paraffin oil affects it. I'm not I'm not completely familiar on the subject. If I remember correctly, DNA is not so well preserved in kitten. Maybe in the future, it's interesting if you have genetic codes of specific species that you're interested in, and you can try to extract it from the sediment actually.
And just from a like a museum sample specimen, I know some people who were able to really extract DNA from some very, very old museum samples from hair, which are also not preserving DNA very well, from teeth, and so maybe this could be an option.
Okay, thank you.
Good, thank you, Nick.
I'm just curious how easy is to identify to species level, because you have like such fragmented remains. It's often like just genus or family, or often you can identify species.
The longer I spend on it, the more is possible. I can now help here in Lithuania. I could help at Kaunas Museum from the curator, who is a great help. If you got more specimens, then maybe you find that one individual is better preserved, and then you can compare. The nice thing is, because it's so well preserved, these insect remains, the hard exoskeleton have a lot of features on it. You got microsculpture, macrosculpture, dots and stripes and everything, which you can then even compare. You can compare small parts with the full habitus of the species. So sometimes it's possible, and in other cases, like half of the times we have to say, okay, we can just know the genus or maybe the family, and that's it.
Yeah, well, that's interesting, because I'm wondering what if it would be possible to create some kind of software that would just automatically analyze the images. Can you like make a database of species? I know, scan them with micro CT, whatever, all the ultrastructure, and then just take pictures of all of these parts, and maybe it's possible to do some automatic classification, and then you don't have to worry so much about if you get it right or wrong. Or
again, I didn't follow this development very closely. I think the development is slow. There's been some tryouts. They managed to identify with software just like species that I caught in some Malay strap a couple of years ago. So it's slowly developing as well. They also are using what's it called, proteomics. They want to develop further to not only insects but any organisms that they don't have to identify it anymore by museum specimens, but they can just run it through a machine by their protein signature. That's what also developed me.
Yeah, that's cool.
Hello. What do you think about trait-based approach? For example, if you have samples from your several sites, maybe you can apply a trait-based method, for example, of some of parts of a beetle that is left there, and maybe you can use this kind of approach.
Could you remind me what is trait-based?
Trait-based. For example, if you have traits of particular species or a group of beetles,
yes,
and maybe you can have a trait database from a site, and you can analyze this between sites. I don't know if you are familiar with the trait
like
trait analysis.
Diet and it's kind of
functional traits.
Well, we try to assert them, but maybe more is possible. So I'm happy to discuss with you further.
I'm not familiar with that. I'm just curious if it's possible.
Well, we look at this in general because many species have a lot of different traits, and we have to make these summary of sites. We also kind of have to summarize everything. Like you got a species with like moisture, but also lives in forest, which is dispersal, and he lives on a certain, he eats fungi, but then you still have to summarize it into just a few things. So get lost sometimes.
I think on this topic, I can offer some guidance, similar to how you would build a climatic niche. Like that, you would do the same thing with a trait-based approach. So instead of using preferences for instance, like temperature preference, you would have different preferences or different categorical variables to say that this beetle is mostly a grazer, and sometimes it can also be a predator or something like this. So there are some various ways to code this information, and the good thing here is that coleopterans are very well defined in the trait databases, and there's an upcoming trait database that I'm a part of that we're trying to combine all traits information from invertebrates at a global scale. So if you need some help with this, I could point you in the right direction, but there definitely are quite large scale trait databases that you could use for this purpose.
Yeah, it can be interesting to try it out.
Mm-hmm.
Thanks.
The next presentation is by Dr. Denis Copilas.
Okay, so we will still now try to come from the past more towards the present. So, but we will go a bit further in the past, a few more millions of years. So this is, I'm actually at the end of my postdoc. Actually, it's my second postdoc here with Asta Audzijonytė. And basically, the aim of this project was to try to understand the evolutionary history of these really magnificent critters. So I will try to explain what amphipods are, what is Ponto-Caspian and all this stuff. So amphipods are crustaceans, but how they stand out among crustaceans is perhaps that they are the most morphologically and ecologically diverse group. I mean, they really come in many different shapes, sizes, and basically they occupy any kind of ecological niche in the water, but some species are even terrestrial or semi-terrestrial. Amphipods have also diversified in freshwater quite a lot. 20% of all known species are in freshwater. They hate the tropics for some reason, so in freshwater they are mainly in temperate regions, and some have diversified into these ancient temperate lakes. So an ancient lake is a lake that it's older than 100,000 years. So there are some radiations in these lakes, but more spectacularly in Baikal and in the Caspian Sea. So yeah, so Baikal and Caspian Sea are the most rich in species, and the subject for this project is mainly the Caspian Sea and this region in particular, which is called the Ponto-Caspian region. So it consists of the Black Sea, Caspian Sea, Azov, and what's left of the Aral Sea, and the lower courses of these rivers that flow into these seas. These seas, they are all remnants of a gigantic, let's say, lake, Parathetis, which is arguably was the biggest lake in the history of the Earth. For most of its history, it was actually a sea, but towards the end, it became a giant enclosed basin. So it stretched all the way from the Alps to the Himalaya. So this area had a quite fluctuating geological history, let's say. This lake would often expand and contract, and chemistry of the water would change very often. And because of this, the fauna that inhabits the area today is very ecologically tolerant. So these animals, fish, mollusks, or whatever, they are environmentally tolerant. So this area has become a source of many invasive species throughout the northern hemisphere, like the zebra mussel and gobies and some amphipods, had quite detrimental effect on local species. Okay, so Among these Ponto-Caspian organisms, amphipods and this specifically this gamma ray group are the most diverse in the number of species, ecologically, morphologically, and so on. It's quite a spectacular radiation. There's at least 90 known species, all of them endemic, 34 genera, and 4 of the 5 families, which are actually not real families, as I will show, they are endemic to this region. So these animals are like from a few millimeters to up to a few centimeters in length. Oh, doesn't work too good. And we have classified these animals into 4 main ecological morphological groups. So ecomorphs, and an ecomorph is a type of morphology which is encountered in a specific type of habitat. So there's animals that we call crawlers. They have like long, skinny antennas and legs. They can crawl easily under stones and hard substrate. There's diggers. These are very robust animals, very hairy, and they burrow themselves into sand. This digger ecomorph is actually quite characteristic for this group. Then there's clingers. These animals sit on plants and algae. They are very well protected. They have a strong cuticle. They have this very nice ornamentation and colors so that they camouflage very well. And then there's a few symbiotic species which live only on other organisms. So in this case, the species lives on crayfish and specifically on the underside of crayfish. And what's interesting in this radiation is that these ecomorphs are not unique to the Ponto-Caspian radiation. These ecomorphs have evolved several times throughout the entire Amphipoda order. So this is a phylogeny of all amphipods, and with colors there we see different evolutionary lineages that have sparked the same morphologies. So this is actually really interesting because we see the same morphology evolving multiple times. It means that evolution is repeatable. So if it's repeatable, it means that it can also be predictable. And if you can predict it, you more or less understand it. So basically, this is one of the main aims of evolutionary biology, is to understand why organisms look the way they look, why they behave the way they do, and so on. So this is a very nice, I would say, model system, this radiation to understand evolution. So this postdoctoral project had 2 main aims. One, we wanted to take advantage of newly discovered fossils, allegedly of Ponto-Caspian amphipods, and try to also study the fossils that were known before in a phylogenetic context. Basically, you wanted to know how these fossil species from 10 million years ago, how they are related to the modern species, and then to integrate this information with molecular data, morphology, paleontology, and so on, using the latest statistical methodologies to try to reconstruct how this radiation evolved through time, how old it is, how it was influenced by the geological history of the region, how ecology evolved, and so on. So with fossils, there's a handful of fossils described like 100 years ago from the Caucasus. These are dated to around 9 million years ago. But a few years ago, I was contacted by colleagues from Romania. I'm a Romanian myself, and paleontologist colleagues that they found fossils of amphipods, and if we could like try to figure out what these things are. So I was like really excited about this opportunity because I'm a biologist myself. I always liked dinosaurs and paleontology, so this was like, yeah, very exciting for me to work on. So I received like probably 100-something specimens. Most of them were actually not very nicely preserved, but a few were actually quite spectacular. So I just did some drawings with a tablet to understand better the morphology. And then if you want to fit these fossils into an evolutionary tree, how do you do that? Because they don't have DNA, like how it's commonly done today. So we did this based on morphology. So we built such a matrix based on morphological traits, and it's like mostly binary coding, so 0 and 1. So for example, you make a trait like antenna, long or short, you code it 0 or 1, and so on. We did this for 114 morphological traits, put it in some softwares that make these phylogenetic trees. Here, I don't want you to bore you too much with the details, but the main idea here is that these fossils, the Romanian ones and the Caucasus ones, they form this well-supported evolutionary lineage. And what is strange about this lineage is that they definitely belong to this radiation of Ponto-Caspian amphipods, but they are not closely related to anything that lives today. So basically, it's just an evolutionary dead end. They existed for a bunch of million years, but for some reason, this lineage just disappeared and without any modern descendants. And it turns out that we did several more analyses, also morphometry, and it turns out that these fossils from Romania, they belong to 2 species in a new genus, which we described, and we published this year a paper on that. So it was a very nice collaboration for me with paleontologists. I really learned a lot. And then move on to the main topic of this postdoc, which is to understand the phylogeny of this group. We did field work in Azerbaijan, in Kazakhstan, and Romania. These are the hotspots of evolutionary diversity of these animals. I also received a lot of material collected by other colleagues in other years. In total, we had 60 species. I tested 13 markers for DNA sequencing. And for the final data set, I settled on 10 markers, which had very good amplification success. Some were very variable, some were very conserved. So it was like a nice choice. So just yesterday, I received the last sequences. So this data set is complete. And I ran a few analyses just to show you what happened. Actually, here we were in Kazakhstan and we were trying camel milk. It's not very tasty, but it was a nice experience. Okay, so with these 10 markers, I put it in a software called Beast, and it makes such a tree. I don't want to, again, bore you with the details too much, but for example, here is like this tree calibrated in time, right? These are millions of years, how we go in the past. And this is the origin of this group. So it's originated somewhere between 14 and 10.5 million years ago. But the lineages that are alive today, the origin is at around 11 million years ago, plus minus a few million years. So this tree also has a lot of green nodes. So these with green have really strong statistical support. So I'm actually really happy about this. There's a few problematic areas here, which means that here, for example, it's very hard to understand what is the relationship between these lineages. It means that there was a very fast, explosive evolutionary event, and it's very hard to reconstruct what happened. Move here. Okay, so how do we relate this to the geological history of the area, right? So let's have a look at the maps, and then I also want to plot here. This shows the rate of species formation through time, so diversification rate. You can see it was not constant, but it fluctuated a bit. So I want to point out that, for example, when the extant species started to radiate, this coincides very well with the greatest extent of the Parathetis 12 million years ago. This is a time when this basin became isolated, more or less. I'm not a geologist, so if I say nonsense, please correct me. So this lake became brackish, and these animals, they really love brackish water. So this is when they started to diversify very, very fast. Then 8 million years ago, there was an interesting phase where the sea really contracted. And during this period, again, we see a really fast rate of species formation. Then at around 5 million years ago, the sea expanded again quite significantly. And again, we see here quite increase in diversification rates, and especially this clade of species appeared, which is very significant, as you will see. And then at around 3 million years ago, the Caspian and the Black Sea basin became isolated. The thing is that they weren't isolated forever, because during the glaciations, what happened when the glaciers were melting, a lot of this water would flow into the Caspian Sea. Caspian Sea would expand massively, and then water would overflow through the Manich Strait into the Black Sea. So there were these brief periods where the two seas communicated and fauna could exchange from one sea to the other. And this is actually, for me, it was of great interest, because we looked at these species pairs. So we have these clades, and they consist of two species or two lineages. One is endemic to the Black Sea, one is endemic to the Caspian Sea. So we wanted to understand, when did they split? Does it correspond to these glaciation cycles or not? So when we plot these out separately, we actually see that very few are older than 1 million years, and most of them are actually more recent than a million years. So this correlates very well with what we know about glaciations, and that especially during the last million year, the glaciations were longer, and the amplitude before interglacial and glacial periods were bigger. So it means that at different times, but these animals disperse from one basin to another, but we see three major episodes, one at 800,000 years, one at 500, and one at 200,000 years. These correspond very well with periods of very low sea level in the Black Sea. Okay, so this is about geography and geology, and now we look about to try to understand how ecology evolves. So I mentioned these ecomorphs. Here I add a few more categories. So for example, these pie charts at nodes, these are reconstructions, statistical reconstructions, to try to understand what was the ecology of these ancestors. So for example, the ancestor of all the group was 50% probability to be a crawler or a clinger, for example. And actually this corresponds quite well with what we know from the fossils. Fossils have such a morphology somewhere in between. So we see that the first splitting lineages, they were mainly these clinging animals. Some also moved to a more symbiotic lifestyle. Then we have this radiation of crawlers. And then these crawlers at some point 5 million years ago gave rise to this digger ecomorph, which is very adapted to burying sand. And what is spectacular here, I think, that this digger ecomorph, some lineages even became less specialized in digging. Some completely lost the abilities. And for example, here in this lineage, not only that it lost the ability to dig, this was actually an advantage for them, and they started diversifying again. So you can see how they can just go back and forth between these ecomorphs during their evolutionary history, which is quite spectacular, I would say. Here, I don't want to bore you with the taxonomy, but I just want to point out some embarrassing situation, because there are these genera, for example, which are not supported as good genera, neither by molecules, neither by morphology, because they do not form like a well-supported clade or lineage. What we call Ketogamarus, for example, just appears in various parts of the tree and so on. So a lot of work has to be done here to figure this thing out. The same with families. These 4 families are endemic, but we see that they do not form a separate group from the Gammarid family, the parental family. They are just highly specialized, ecologically highly specialized. Because of this, they also have some peculiar morphologies. And people, when they first found these animals 100 years ago, 50 years ago, they said, wow, this is so peculiar that it has to have its own family. But neither morphology, neither molecules agree with this, so these families have to be scrapped, unfortunately, but that's how it is. And this is just some extra stuff that I didn't plan in this postdoc, but it turned out that we managed to do a manuscript where we describe 2 new species of the genus Lithogamarus from the Caspian Sea. So this is it. Thanks for your attention. I leave you in the company of some nice images from Kazakhstan.
Thank you very much for your very interesting presentation, especially with phylogenetic analysis. It's very impressive. Prašau klausimų. Questions, please.
Thank you for presentation. As far as I understood, sorry, I am not a biologist. This is shrimp species from Ponto-Caspian, all of them brackish water species, or fresh water also, because it was indicated with meltwater basin during the glaciation.
Yeah, so they are actually brackish water species, but they are very tolerant. So a lot of them can occur in fresh water. Many of them are actually invasive in Europe, so where they just live in fresh water, so they are quite tolerant.
So next question, maybe you have any information about such Ponto-Caspian species in the Baltic basin, like invasion species, like there is an polymorpha that we, according to our understanding, is an invasion species that was transported from this basin to the Baltic. It's only problem when it happened. We have some controversial data. It may be possible to find such species in the Holocene sediments in post-glacial.
Yeah, I think it is possible. I think that these species might have spread from the Ponto-Caspian basin not only now, but in some previous interglacial stages. But unfortunately for amphipods, this is very hard because they barely fossilize. There very few fossils are known. So, but for example, there are Ponto-Caspian, I think, mollusks from the genus Dreissena, these bivalves, and I think in Lithuania there have been, or in the Baltic area, found like these fossil or semi-fossilized remains. So because for me, I think it's perfectly logical that these species would expand in the interglacial, because there's a lot of ecological space for them in Europe to spread, because the native species in Europe are quite limited ecologically. You have many species, but they are all very similar ecologically. So there's a lot of this niche space, these vacant niches that these Ponto-Caspian species occupy. And this is also some other thing that I'm doing. And I can tell you now that these Ponto-Caspian species occupy completely different ecological niches than the native species. So basically they have a lot of room basically in the environment where they can just settle. And
Thanks, Dennis. Thanks for making it clear. Presentation was good. You mentioned that the gamma families that are currently used are not supported by molecules or morphology. Do you have a recommendation? I guess that's quite a broad question, but do you have a recommendation of how these gamma should be classified? And are they all just the Gammaridae, or are there some other groupings that form?
this is actually a question we are all facing with in this Gammarid group, because this Ponto-Caspian group, and also like in Baikal, these radiations are nested in bigger clades, but these clades are very morphologically limited. All these species are very similar. But when they get into these ancient lakes, they really explode in all this diversity of forms. So if you want to classify it by morphology, okay, you can do it, but in an evolutionary context, all these families, they don't make sense. And the point of Linnaean taxonomy is that it has to reflect, the evolutionary, the process. And so this is, I don't know, we are still scratching our heads, because if you can synonymize these families, but then the Gammaride family would be something which you cannot really define, like to give a diagnosis, what is a Gammaride, because it's just crazy amount of diversity. So I don't know. This is still something that we are trying to figure out if it's possible.
Around as a sea. And you showed very nice picture. So now it looks like fragmented lakes, connected and not connected. And this happens very rapidly in the 50 years. Do you see dramatic changes in fauna of your objects?
From Aral, yes. I mean, all of the species, at least of amphipods, disappeared from there. But the historical data that we know from there, so before this disaster happened, there weren't many species, maybe a couple of species, because it's quite shallow. And until recently, it was basically connected to the Caspian Sea. So I would say that it wasn't such a big disaster for them, because these species that live in Aral, they also live in the Caspian Sea. So,
and let's move from the past to the present. And the fifth presentation by Dr. Giedrius Šidlauskas is dedicated to molecular investigation of Toxoplasma and Sarcocystis parasites.
Hello for everyone. Today I will be sharing you with our plans for a study focused on two key parasites. It will be Toxoplasma gondii and Sarcocystis species. These parasites which can affect both wildlife and humans, and are growing concern in zoonotic disease transformation. So we far successfully collected samples from beavers across Lithuania. And our next step is to expand this research to include small mammals like mice, voles, and rats. Using advanced molecular techniques, we will test heart, brain, and internal organ samples for these parasites. Our lab is fully equipped to perform DNA isolation, PCR, and sequence to identify and analyze these pathogens. So let's move on with the simple results. And now I introduce that small mammals, particularly rodents, play a key role in spreading pathogens like Toxoplasma gondii, which is a serious foodborne disease. While studies of the genetic diversity of Toxoplasma gondii in rodents are limited in Europe, none have been conducted in Lithuania. This research will focus on identifying the strains of Toxoplasma gondii found in rodents, as these animals can act as sentinels by ingesting ecosystems from environment, which could help us understanding the risks these strains pose to humans and the animals. The fact that this has not been studied yet presents a serious gap in understanding the full scope of zoonotic risks posed by rats. If Sarcocystis are present in wild rats, they could serve as a major source of contamination in environment, affecting other wildlife, livestock, and humans. So rodents are incredibly diverse group of mammals, making up about 40% of mammal species. Their widespread presence across various environments means they interact with both wildlife and humans populations, increasing the potential for disease transmission. This project aims mainly to develop molecular techniques for accurately identifying Sarcocystis species, using small mammals as their intermediate hosts, by focusing on these animals, which often serve as a key part of the parasite's life cycle. We can improve our understanding of how a Sarcocystis species spread and potentially cause disease. Developing these molecular methods, we will enable more precise decision and contribute to better control of these parasites. This project will estimate the occurrence identified by molecularly characterize Toxoplasma gondii and Sarcocystis species, which are protocysts and parasites from the family of Sarcocystis in rodent populations. By analyzing these parasites in rodents, we aim to gain insights into their prevalence and genetic diversity, contributing to a better understanding of risks they pose to human and animal health. We have some objectives, mainly to identify and first of all to find these pathogens. So we will take techniques of genetic diversity. So and we will investigate in this in small mammals and the bigger ones in the beavers. We have a great laboratory, which is great equipped with comprehensive and advanced instrumentation, including facilities for DNA isolation. So this is well established infrastructure ensures the capability to perform cutting-edge molecular research, contributing significantly to advancing knowledge in the field of molecular ecology. First of all, we have to trap small mammals. So and then we will be identifying them. We will have to wait and measure. So following this, the individuals will be detected by determining their age and sex. Wolves will be identified by the species level based on dental characteristics, while other species will be visually identified. The intestines of round rats will be examined for Sarcocystis species using both microscopic analysis and DNA extraction. So the primary focus will be on the zoonotic and pathogenic Sarcocystic species. And the detection of genotyping of Toxoplasma gondii will be performed on brain and heart samples of collected animals using pepsin digestion. Nested PCR, which targets in ITCA first region, will be applied for this high sensitivity in detecting parasites. Positive samples will then undergo further analysis through Sanger sequencing, and genotyping of Toxoplasma gondii from beaver brain isolates will be conducted using microsatellite analysis. So we have some software. Our project involves capturing around 400 small mammals, like mice, wolves, rats, in 2 different environments, meadows and near a homestead across Lithuania. We will use standard methods to capture them, and we will use traps. And additionally, 100 beavers we have to catch. So it's quite risky tasks. We have not much time because the hunting season is quite short. So we have to do the best to capture them. Also, we have planned some internship in Madrid University or Salavet Laboratory with Professor Luis, and they are like professionals in parasitical methods. And the first results, these are results, not results, as you can see, but we have found some interesting things in beaver brains. So I will show you. So we have samples we have tested 81 samples, and from the first start, from the first tested samples, we have 3 beavers which they were like positive, and we can identify, but not always from the first time we can identify. So we have to make it another time. So 11 beavers from 81, they are positive. So it means like around 10% of beavers can be sick. So Toxoplasma, comparing to other countries, our 10% from these results is quite low percent, because like for example, in Denmark, we have 30% of beavers that they have a Toxoplasma. As I mentioned, it was the best part and the worst part by collecting beavers. It was quite difficult because they only show in nighttime, or you have to have some help, or so the help is from the dogs, and you know, it's quite difficult and interesting to get them. I am a hunter, and the background is a forester, so beavers and trees, they're not together, because beavers like they can change the psychology really strong. So we have managed to collect beavers by gathering hunters. For example, in one hunt, we have gathered 200 hunters with like 50 dogs, and it was like really great organized hunting, but it's quite crucial, you know, like 200 hunters gathering and coming with beavers, but for the studying purposes, it was really nice, and we have really great experience there. And so some results are in popular magazines. We were discussing with the hunters and with the society. We have written some articles in journals to inform hunters and the society that Toxoplasma can reach them by making snacks from uncooked meat, you know. So this is the purpose of this research, is really have many poses that we can reach humans with these results. Also, we have made some media, and we were also with that hunting in the news. So the whole Lithuania knew that we have this research done. And also the research is continuing. So by the last slide, I would like to ask you, maybe you have any rats to share with them. Because it's really, I thought that beavers will be really difficult to catch, but I have never imagined that catching rats is impossible. Thank you.
Ačiū labai už jūsų pristatymą. Kokie būtų klausimai? Questions, please.
I am just curious, why beavers, not foxes that are closer to humans and animals?
Thank you. So you mentioned that humans can get infected by eating beavers. Was that the idea? So how will humans get infected by these small rodents? Yeah, how is it transferred from these small rodents to humans? By the cats. Okay, so the cats eat these small rodents, and then. Okay, so it's in the end it's cat shit. That's okay. Mm-hmm. And I remember from the Netherlands, there were a lot of rabbits with toxoplasmosis, I think, and they were blind and they were very like external features you could see from them that they had toxoplasmosis. That's not the case with beavers or these other small rodents.
So you can find this.
Okay, so it takes quite a while before this becomes externally visible. So that's why you should still catch them and dissect them.
Yeah, of course. You by limiting the population, so you can find it better, because if there are too many like individuals, so diseases are spreading like widely.
Is it necessary to kill the beavers to identify Toxoplasma?
Yes. You know, we have gathered 170 beavers, as we plan to catch 100, but from that 170, like around 100 is okay with the brains, because when you hunt, sometimes it's impossible to have all the samples.
Also, you mentioned that trees and beavers have an effect on some forest ecology or something like this. But it's well proven that beavers have like a super positive effect on rivers.
Yes.
So, like, what is the justification there? How can you justify killing beavers because they affect forests but not rivers?
For ecological aspect, beavers are like great builders for the ecological niche and structures, but mainly they cause some problems for the foresters, also farmers. They build dams on the on the shores, so it's impossible to compare, you know, ecological aspects with a business or something like that. I'm not against the beavers. I like them, but for the studying purposes, so it's great material.
Okay, thanks.
I have a quick question. So this 100 or 170 beavers, where they were collected, in one or several regions in Lithuania or across the whole country? And you have processed some of the samples already. How often do you find beavers infected with either Toxoplasma or Sarcocystis? Thank you.
We have, at this moment, we have found 10% in infected Toxoplasmosis beavers, and we have catch them or gathered from the whole Lithuania, starting from Zarasai region, ending by Klaipėda shore in the middle of the Lithuania, here by Vilnius. We have a great community, as we were like building our hunting hunters, and we were informing them in the journals, in media, in news. So they collected and gave some samples. But at that moment, the spring period was really difficult. I was days in hunting, and in the night I was skinning them, preparing, and then again. So.
I have one short question concerning the geographical scale of investigations. You emphasized just Lithuania, but I don't think beavers or rats follows state borders. It's impossible. What is your opinion concerning possibility just to take some extra samples or as a control samples from surrounding areas?
By surrounding areas, we were discussing with Latvian hunters. It was one interesting thing that they found one beaver with trichinellosis, not only with the Toxoplasmosis. So we have gathered some samples also, like excluding this research, by taking some samples for trichinellosis also. So samples from Poland, Latvia, and Lithuania will be gathered also. So maybe we will find some interesting things.
Thank you. Evidence that your presentation was interesting for more of us. And the more questions during discussion panel. Thank you very much. The next presentation by Alžbeta Šujanova, Exo-erythrocytic stage of Hemoproteus parasites in black caps, confections, and the challenges in identification. Supervisor, Dr. Gediminas Valkiūnas.
Dear colleagues, my name is Alžbeta Šujanova. I'm from Laboratory of Parasitology, and my supervisor is Dr. Gediminas Valkiūnas. And in the next few minutes, I will share with you some of my results during my postdoc project. I would like to mention that this project started in January of this year, so I have been working on it approximately for 8 months. First of all, I would like to tell you some very important 4 pillars to better understand the overview of this project. The first one is Hemoproteus. What is Hemoproteus? Hemoproteus is intracellular blood parasite. So these parasites could infect birds. If you know human malaria, it's a very dangerous disease caused by Plasmodium. And Plasmodium could be also found in birds. So Plasmodium and Hemoproteus are very similar and familiar parasites. They are transmitted by blood-sucking vectors, some insect, and this parasite could be associated with different groups of animals, not just with the birds. The second very important term is exo-erythrocytic stages. What is it? I'm not going to overwhelm you with a very difficult life cycle of this parasite because it's not necessary for this presentation. But since we consider this parasite as a blood parasite, they have one development stage which is not connected with blood itself, but with the tissue. And here you can see some some very nice pictures of this development stage in the tissue in the birds. So I'm focusing on this stage of the development, yeah, not in a blood stage, but in the tissue stage. Third one is the host, Sylvia atricapilla. Why we choose this host? First of all, Sylvia atricapilla, or Eurasian blackcap, it's a very common bird in Europe. And every year the ornithologists found higher and higher numbers of these birds, so which is very good. During my PhD in Slovakia, I found out that 70% of these birds were infected with some species of Hemoproteus, so the prevalence is very high. And also this bird have a specific parasite, which is very good for me to better understand the pattern of parasitemia in general. And the fourth is the challenge. What is the challenge of this project? So as I told you, the Sylvia atricapilla has some specific parasite. And when we are talking about Hemosporidium parasite in general, we are talking about the morphospecies and the lineages, and they are two different terms. The morphospecies is connected with morphotype. So basically, when you take a look to the microscope, what you can see, yeah? In a Sylvia atricapilla, you can see three different species: Hemoproteus parabellopolski, Hemoproteus pallidulus, and Hemoproteus homogenea. But when we are talking about lineages, we are talking about the DNA, about molecular analysis. For instance, Hemoproteus parabellopolski has at least 4 different lineages. And these lineages are very similar, but they are different just by 1 or 2 base pairs. So they are different in 1 nucleotide. So yeah, it's very challenging to find single infected bird by morphospecies and by lineage also, because Sylvia atricapilla, it's highly prevalent bird, but very often, I mean 95% of birds are found in co-infection. So it's very challenging to design some specific probes, specific primers. And when you are working with this parasite, you want specific primers, you want specific probes. So this is the main challenge, to find a single infected bird and work next. So now you're familiar with the main ideas of this project, and now we can move to the material and method and procedure. So how am I working? First of all, I need to identify birds infected with some young stages, young blood stages of Hemoproteus, and how I'm doing this. So my field work, it's located in Ventes Ragas. Probably most of you have been there. It's a very nice ornithological station. This should be 2 videos, but somehow it's not working. So you can imagine how we are taking blood from the birds. So during this fieldwork, I'm doing very quick microscopy and trying to find a suitable host for my project. And what is the suitable host? So first of all, the bird has to be infected by Hemoproteus. The intensity of infection should be also not very low, not very high, and it should be infected with young blood stages. If I found such a bird, I euthanize the bird, and yes, it's necessary to kill the bird. It's sad, but it's necessary. And then I can move to the laboratory. And first, I will do some nested PCR to determine the specific lineage of the bird. And now I can move to the most fun part, to the histology. For histology, I'm using 2 different methods, the SISH and the H&E. You can see they are similar, but still different. The SISH, it's chromogenic in situ hybridization. It's combination between molecular method and histological and microscopical method. These videos, it's not the Hemoproteus parasites, but they are from my previous work for Isospora parasites, and we are looking at the intestine of the bird. And all these black dots, which you can see, they are parasites, which is very good for me that I know for sure that, yeah, this is the parasites. And then you can compare your second slide, the H&E, hematoxylin eosin stain slide, and tell, oh, yeah, this structure is actually parasite. And then you can describe the morphology of the parasite. So combination of these two methods, it's very crucial for this project. So what are the goals of this project? The first, to confirm these exo-erythrocytic stages in tissue using the SISH method. Then design some specific probes for the specific parasite, which I introduce you. And finally, determine some pattern of organ tropism between single infection and co-infection for the first time, because, yeah, the co-infection is quite tricky. So, about the research progress. The first stage of the project is already behind me. I collect all the histological material. The second stage is right now. I'm trying to analyze all the samples by PCR and also histologically. Then I will try to design some specific SISH probes and identify the co-infection for the first time and publish the result. So now we can move to some awesome result. So far, I analyzed 29 birds. During this September, I was in fieldwork and collected more birds, but these birds aren't in this presentation. So my results overall are composed from three main groups of the results. First is microscopic observation of the blood smear, then the PCR and molecular results, and then histology. So during the microscopic observation, 28 birds from these 29 were positive for blood parasite. The one bird was found dead, so we weren't able to tell if it wasn't or wasn't infected by blood parasite. And on this plot, you can see that just 6 birds from this 29 was single infected with Hemoproteus parabellopolski, which is 90%. 22 birds were co-infected. So most common co-infection was between Hemoproteus parabellopolski and pallidulus, which is 39%, but almost half of the birds were co-infected by different combination. So yeah, it's tricky. So remember this number: 6 birds from 29 were confirmed to have a single infection. So from these 6 birds, just 3 birds had single infection confirmed by PCR. Yeah, so yeah, we have just Hemoproteus parabellopolski, but then you see, okay, but we have lineage C1, C2, C11, and what now? So 9 samples had co-infection, but I was able to identify just the dominant lineage. For 11 samples, I wasn't able to identify nothing, and 6 samples were tested negative. This could happen because if the intensity of infection is too low or too high, the PCR just doesn't work well. So you have to repeat the samples and play with it. So it's tricky sometimes. And now the histology. 7 birds were positive for SISH, 9 for H&E. 1 bird is still questionable, and I found also the blood signal in 12 birds. So in heart, I found some merons and megalomerons in 3 birds. For one of them, the middle one, it was confirmed also by SISH, and by the 2, I didn't find the SISH signal, but I can tell that it's Hemoproteus parasite because I have a great colleague and she helped me. In 2 or 3 birds, I found infection in gizzard. Gizzard is part of the stomach. 2 of them were confirmed by SISH, and this 3rd one, it's questionable because I'm not sure if it's parasite. If you have any guess, please share it with me if you think or no. Also, kidney was infected in 3 birds and the mussel. So as you can see, for those 2 pictures in gizzard, they look similar, but the rest, I don't see any pattern, any morphological pattern to distinguish which morphospecies it could be. So again, if you have any guess, just share it with me. And I'm working also with this blood signal because I told you it's Hemoproteus is a blood parasite. We have blood in our whole body, so also in the tissue. So sometimes I can find some very small black dots, which is the blood stages in the tissue. And it's very interesting if I can see these blood signals just in one organ or different organs, or one organ is more infected than others. So I'm working also with this results. So overall, I have 5 different groups of results: the blood smear, PCR, CISH, AGNE, and blood signal. In a perfect universe, I would find every step positive, but we are not living in perfect universe, of course. So sometimes I found the blood smear and PCR positive, and also blood signal. Sometimes I found also tissue stages, but not confirmed by CISH. And sometimes I found just the blood stage. So yeah, as you can see, it's very tricky and challenging project. So wish me luck. And what next? In the next few months, I would like to focus more on 18S gene and try to design some specific probes to target the specific parasites, and just hope it will work. So yeah, that's for now. Thank you for your attention. If you want to follow me on LinkedIn or Instagram, I would be happy. Sometimes I post interesting posts. So thank you again.
Thank you very much. Questions, please.
Regarding these co-infections that you see in the chromatograms, we had similar problems with this crustaceans I work with Microsporidia. And what we did at some point, we sequenced these PCR products using next generation sequencing.
Mm-hmm.
We use nanopore in this case, and like it was very nicely, it can really distinguish between all these variants. Or you can use Illumina. Actually, I think we even have nanopore here at the Institute. So it can be tried, and then based on these sequences that you get, you can then really design specific primers for these lineages. And I think
yeah, because
could
actually
be something to think about.
Yeah, actually, next generation, it's not very often used with Hemosporidium parasite, but I was thinking about it to try something else. Yeah, so maybe I will.
Because you can do this just on the PCR products. You
Yeah.
know, there you have 2-3 peaks in different places, and then just sequence that.
Mm-hmm. Thank you.
In this case, it's contaminated with bird DNA. You see, a lot of bird DNA. So this is the Macrosporidia. You can take clear spores without vertebrate DNA. Here it will be a lot of DNA. So it's not so straightforward.
You do a PCR, right? And you have these specific fragments. You have also from the bird, from the parasites. But when you sequence, you just sequence everything. And then when you reconstruct all of this statistically, then it tells you that there are like, I don't know, 3 or 4 amplicons. And then you just blast them and see which is the bird, which is one parasite, the other. And then you could try to see if it's possible to design these specific primers.
We'll try. Yeah, thank you.
Thank you, Betty, for your presentation. I would like to ask, how is your project fitting in the knowledge of the exo-erythrocytic stages that are known already, you are working with a different bird species. Do you expect to find some differences in exo-erythrocytic stages of Hemosporidian from the previous study? For example, maybe morphologically, they can be different or located in a different organ. So what is the novelty that you expect to?
You know, the main idea of this project is actually this co-infection. That's why we choose Sylvia atricapilla, because very often it's finding co-infection. And yeah, Melanie and also Melanie Tchoumbou, they are working with these stages. But for the first time, we want to find bird for sure co-infected with different morphospecies in one individual, and try to find out if it's some organtropism or something, or if this tissue stages somehow cooperate. Yeah, so this is the main challenge.
The co-infection.
Yeah, understand the co-infection in tissue.
Okay, thank you.
Next presentation by Melanie, Dr. Melanie. I invite you to the step. Exo-erythrocytic development of little investigated avian hemoproteosis, supervisor Dr. Gediminas Valkiūnas.
Hello, everyone. Thank you, first of all, for giving me the opportunity to share some results of my postdoc project. My project was founded in 2022, and the main aim is to understand actually what happened in bird organs during infection with Hemoproteus parasites. Again, Hemoproteus parasite. So first of all, I'm going to share with you what is actually Hemoproteus and why we are interested with this parasite. So Hemoproteus parasites are a diverse apicomplexan parasite belonging to the order Hemosporida, family Hemoproteidae. They are transmitted to birds and other vertebrates by the bite of midges and loose flies. Till date, 179 species of Hemoproteus have been described morphologically, and more than 2,000 lineages reported in Malawi database. For all these existing species, only the exo-erythrocytic stages have been documented for only 14 species and 21 lineages. This shows the big gap in knowledge regarding the exo-erythrocytic stages of Hemoproteus parasite. And this is one of the reasons why we are interested in this parasite. In general, the life cycle of Hemoproteus parasite involves three main stages: the sporogony in vector, the exo-erythrocytic stage in the host, and the erythrocytic stage in the red blood cells. Previous studies have shown that during the exo-erythrocytic stage, merons or megalomerons developed and can damage many bird internal organs, including the brain. Research on bird histopathology is still in its infancy, and to contribute additional knowledge in this field of research, we sample bird Adventist ragas ornithological station using different trap techniques. And in total, 8 infected individuals of African migrant Luscinia were included in this study. It is important to note that this bird species is least concern based on the classification of EUCN. Prior to the selection, blood smears of each captured bird were prepared, stained with GEMSA, and observed under light microscope for parasite identification and parasitemia. Based on the results obtained, a limited number of infected birds were decapitated, and blood was collected in set buffer tube for molecular analysis. On the other hand, all bird internal organs were collected and kept in formalin in the field. Once in the laboratory, the organs were processed and embedded in paraffin blocks. Thereafter, several sections of each organ were mounted on slides and stained using hematoxylin. Additionally, a chromogenic in situ hybridization technique, which target the 18S ribosomal RNA of the parasite, was used to confirm any blood stage found. Based on microscopy, all the birds were infected with Hemoproteus attenuatus, and analysis of partial cytochrome B gene revealed that this parasite belongs to the lineage Robbins 1. Histological examination of the infected bird shows presence of merons in the lungs, in the liver, in the pectoral muscles, and in the heart. This result was confirmed after in situ hybridization using a lineage-specific probe. In fact, it's already from previous studies, some researchers could already design some lineage-specific probes, which can be used to confirm infections in tissue stages. We found that lungs in these birds were the most infected organs, and each meron, the size of each meron range from 7 to 47 micrometers. Also, the merons were oval, elongated, and without visible envelopes. In addition, the merons appear at different stages of maturation. We found the developing merons characterized by the presence of cytomers, and mature merons which were full of merozoids. Interestingly, in one of the 6 individuals infected, we also found megalomerons of Hemoproteus majoris in co-infection with merons of Hemoproteus attenuatus. And again, this was confirmed by using a lineage-specific probe during in situ hybridization. The gizzards were the most infected, and the diameter of megalomerons were quite bigger compared to merons. Also, Megalomeron had visible capsule-like envelopes, which differentiate them from merons. We also observe severe inflammatory reactions around Megalomeron in the giza. And the presence of these reactions indicate the virulence of this parasite. So in addition to the different maturation stages observed, empty capsule-like envelope was also found in the giza of one particular of this particular bird. So in normal circumstances, presence of empty capsule-like in the giza indicates that merozoid in this host cell fully developed, gave rise to unicellular merozoid, which were released in the circulation to initiate the development of gametocytes. However, after intensive search in the blood smears of this particular bird, no gametocyte of Haemoproteus majoris was found. Instead, we observe in the plasma a multinuclear structure, which we assimilate to a syncytia of megalomerons. So the presence of empty capsule-like wall in the tissue stage of this bird associated with presence of syncytia provide evidence of abortive tissue, of abortive development. So from our knowledge, to date, there is no documentation of gametocyte of Haemoproteus majoris in thrush nightingale. At the end of our investigation, we conclude that Haemoproteus majoris lineage Robbins 1 and Haemoproteus attenuatus Robbins 1 and Haemoproteus majoris lineage WW2 share different clade. And although these two parasites are not closely related, they both develop tissue stage of in the thrush nightingale Luscinia bird. Also, Haemoproteus attenuatus develop only merons with the apparent preferred location in the lungs. Haemoproteus majoris develop only megalomerons with marked damage in the gizzard. And megalomeron of Haemoproteus majoris was shown to abort development in thrush nightingale. And this is, from our knowledge, the first case of abortive development during tissue stage in a naturally infected bird. Actually, I am at the end of my first year postdoc, and so far, I could contribute to publication of 3 papers. I also attended to several workshops and conferences during which I could learn new molecular techniques, improve communication skill, and increase international collaboration. So, given the huge gap in knowledge regarding bird histopathology during Haemopotus infection, we submitted a new project to further investigations on avian hemoproteosis. I am grateful to the Research Council of Lithuania, Nature Research Center, my supervisor and collaborators. Thank you for your attention.
Thank you, Melanie, for your presentation. 1 or 2 questions, please.
Why do you think that this case, this abortion, how to call it, happened? Like, it's because this parasite infected the different species that it usually infects, or I mean, just any speculation or whatever. It's just interesting.
Okay, thank you. First of all, abortion is when the parasite failed to develop in a particular host. So in this case, based on previous finding, no report of this particular parasite, no report of gametocyte of this particular parasite. We have no report in this particular host, which somehow shows that, in fact, when we found parasite in the host, usually during the life cycle, the parasite should follow all the different stages, which means from vector, it should develop into tissue, and it should be found in the blood stage. So in this case, until now, there is no report of this parasite in blood stage. And also, since during our investigations, we found in the blood tissue a megalomeron, an empty capsule-like wall of megalomeron. It suggests that normally the gametocyte should be present in the blood smears of these particular bird individuals. But after intensive search in the blood smears, we couldn't find any gametocyte first. And secondly, we found this structure, which is likely a syncytia. And also, it's true that actually investigation on abortive development is not yet advanced. And this is the first case that we observe in a naturally infected bird. But when we compare this syncytia to because there is one previous case reported in a captive bird, and we could relate this structure to what was previously published in bird in captivity. So actually, it is quite tricky to confirm with certainty that this is a syncytia, since it's a first case. But we believe it's important to raise attention of researchers that when they found such multinuclear structure in the blood smear, especially in the case where there is evidence of tissue stage with presence of empty capsule-like wall, definitely it raised attention, and probably we think probably in this bird, Haemoproteus majoris, does not complete its development.
Thank you once more.
Thank you.
No questions, only during coffee. And now we move on to future insights together with Dr. Nathan and his presentation, Lithuanian freshwater biodiversity, spatiotemporal trends, and future insight. Supervisor, Dr. Vaida Šeirienė.
Hello, everyone. It's nice to see many people here today. Last postdoc conference, there were very few of us. I think most of us were the postdocs presenting. So thanks for everyone for being here. Today I'll give a bit of a hodgepodge of what we've all spoken about today, because I touch on Lithuanian landscape, I touch on parasites, I touch on ecology. So it's more holistic than these very specific studies that we've seen today. And I'll be looking at freshwater diversity specifically and how I've seen it change over time. So where this all began was at the start of my PhD in 2018. There were really many studies coming out that were showing large-scale declines in insects, specifically from terrestrial environments. And the top part of this figure is from terrestrial environments. But if we look at the bottom part, we see a little bit of a different picture. So in the terrestrial environments, we saw really strong declines in richness and abundance of insects through time. But when we look at freshwater environments, so this bottom section here, we actually see the opposite pattern. So actually freshwater communities are not faring as poorly as the terrestrial communities. And this begs the question, what makes freshwater communities different? I had a few slides that I actually removed that I wanted to talk about scale. And it's important to keep in mind here that when I talk about scale and when we look at these long-term patterns, we really need to think about at what level we're doing our sampling. So you can sample a site, for instance, or you can sample an entire region, or you could sample the entirety of Europe, for instance. And depending how you move up or down in scale, you may get a different picture. So just keep this in the back of your mind. And also the scale can occur on a temporal basis. So if you're sampling in, let's say, 2010 and you're sampling in 2023, you may be collecting different organisms just because of some cycles that are taking place in the background. A lot of the scale can also come about by humans. So humans interact with the environments in a completely novel and different ways. And depending where you are in the environment, these impacts change the scale at which factors are influencing the communities at this place. And this is just a diagram of rivers and why they're so important, because they have so many more so many more factors at various different scales, and they combine all these factors because rivers all run towards the ocean. So, for instance, agriculture on the top right there may be influencing terrestrial ecosystems at one scale, but then they're flowing into the river, and these pollutants and these impacts are being accumulated downstream as you move toward the ocean. So this is what we, with a previous working group of mine, what we tried to look at the pan-European scale. So we saw this increase in biodiversity in freshwaters, and we wanted to check whether this pattern was really ubiquitous across Europe. And in fact, the first two papers to come out of this, one was lucky enough to be published in Nature and another one in Nature Ecology Evolution. And basically, the main result of the Nature paper is that freshwater biodiversity from around 1980 really was increasing. So humans began noticing that we're making an impact on the environment, and we started putting some laws in place. Water Framework Directive came about. Targets for pollution emerged, and we really saw that freshwater biodiversity rebounded from this. However, around 2010, I can't really point, but around 2010 on the left-hand side of the graph and also on the right, we saw a deceleration of this recovery. So no longer were communities continually recovering, but at some point they reached a plateau and they weren't declining, but they were just stopping to recover. So this is very important, is that this is not a decline or a reduction in biodiversity, but just a deceleration. And then we wanted to look at, from a water quality perspective, what's happening in Europe from a water quality perspective. And we see a very similar pattern on the left-hand side of the lower graph, is that we saw this increase in water quality up until around 2010, but then again a plateau and maybe even a slight decline. And on the right-hand side, you see Lithuania from around 2010 to 2020, the data that we contributed. Lithuanian water quality did not increase or decrease, it remained relatively stable. And unfortunately, that doesn't make it good. So 63% of Lithuanian rivers fail to meet good ecological status. And more needs to be done, not just in Lithuania and Europe, because the European average now is 67% of European rivers do not reach good ecological status. So the whole premise of my postdoc was to understand, okay, what's happening in Lithuania? And I'm going to quickly go through the results of this because I presented them last year. But this was the first paper that came out. And here we used 41 sites across Lithuania. And these 41 sites had good representative sampling that had occurred between from 2010 to 2020. At each point here on the graph represents a time series. So in fact, I have 41 time series. And these time series altogether had around 250,000 invertebrate individuals, representing 484 taxa. And taxa here I'm using as a collective term because not all individuals are identified to the species level, but some to the genus and also family levels. We were also lucky enough to have some linked environmental data for 28 of these 41 sites. And I'm extremely grateful to the Lithuanian Environmental Agency and Vienna for really graciously providing this data, because without this data, it would not have been possible to look at these. So I took this taxonomic data, this biological data, I took the environmental data, and I did some statistical magic, and I will present it next. But just bear in mind that talking about scale, it's difficult to compare two sites that are very far apart in time and space. So instead, what I did is I calculated how biodiversity had changed at a single site, and I repeated this for 41 sites. And then I used some Bayesian meta-synthesis models to combine all of these trends and look at an overall trend in Lithuania. And what I will present next is an overall trend, but I'll explain the graph. But first, let's look at how the environment has changed in Lithuania from 2010 to 2020. So flow actually reduced. So there's less discharge in Lithuanian rivers over this 10 or 11-year period. Temperature increased by around 0.5 degrees Celsius. And to us, that doesn't sound like a lot, but if you're a little creature who depends on the external temperature to regulate your own metabolism, this is quite a big jump, at least within a 10-year period or 11-year period. Specifically, also ammonia products, so nitrates or nitrogen, decreased in Lithuania over the same period, but I don't show it here. In fact, phosphates and some toxic nitrogen species actually increased. So while we're doing some things right with nitrogen, some other toxic species are not doing so good. So just keep in mind that there's some climate-related signals, and I group these into flow and temperature, but then there's also some land use-related signals, and this is agriculture. Well, I'm running out of time already. So let me just go through this briefly. So the blue part of this graph, these are positive trends. So of these 41, these are the trends that had increased through time, and the orangey yellow part are trends that had decreased through time. And this black dot in the middle, basically this is the meta-analytic trend, so the overall trend across Lithuania. So in general, across Lithuania, richness and abundance increased over this 11-year period, which is a good thing. Recovery has not stopped. And this is based on the entire community. However, when we look deeper into the changes in specific taxonomic groups, indicator species, so EPT at the top, these are Ephemoptera, Plecoptera, and Trichoptera. These indicator species, although also show positive meta-analytic trends, actually declined at 50% of the sites in Lithuania. So it means that at 50% of the sites, these EPT were doing well, but at 50% of the sites, they were not. But the big takeaway here is that insects and so insects, including EPT, were increasing at a much slower rate compared to crustaceans, mollusks, and annelids. And this was surprising since during post-glaciation, insects comprise a very large proportion of communities within Lithuanian rivers. And now that crustaceans, mollusks, and annelids are really increasing at very dramatic rates, this may beg the question, you know, what are the identities of these species? And it also could mean that some invasive processes are taking place. What's driving these stuff? Again, we look at climate-related drivers and also agricultural-related drivers. So the decrease in flow actually had a negative impact on abundance and richness. So they're negatively correlated, meaning that reduced flow is correlated to a higher abundance and taxonomic richness. And this could be related to maybe what I mentioned before, mollusks and annelids increasing in their abundance because they don't really like very fast flowing water. So some organisms like it, some organisms don't. And these climate-related and temperature was a big driver of abundance and richness increases through time. But agriculture also had a negative impact, saying that these toxic species potentially have a large impact on the organization of the community. So we may see increases in species richness and things like that, but because the community is now shifting to something that is different from what it was before, climate was a big driver in this case. So just to sum the section up, Lithuania's freshwaters are still recovering, although some sites are not doing as good as the others. Larger rivers showed stronger gains, and they're also more represented. So smaller rivers need to have more sampling effort. And since 63% of Lithuanian rivers still fail to meet good ecological status, we need to be doing more. We need to be really considering how we monitor and what policy is put in place to try and curb this stagnation of recovery through time. Climate and agricultural drivers still remain strong, but according to a new report that was released on Tuesday, some emerging pollutants, specifically bromulated flame retardants, these are really, really important, and they hold a very detrimental impacts on biodiversity in Europe. So now the second part of my talk, I will talk about dipterans specifically, because my postdoc is focused on how changes in biodiversity may change biodiversity. That's the title. And here we're looking specifically at dipterans, which are vectors for the Hemosporidian parasites that we heard before. Spoiler alert, dipterans also increased, a bit lower magnitude compared to the other groups, but dipterans increased. And on average, at each site within Lithuania, each site gained 1 taxa and around 21 individuals. So this is just some backward transformations. And then we asked, if there are changes in these dipteran communities, how would they impact these Hemosporidian communities? So there's 2 hypotheses. One is a null hypothesis that when you have a greater diversity of dipterans, you would have increased prevalence and diversity of parasites. The alternate hypothesis would be that there's a greater diversity of vectors, and because there's more vectors and parasite dynamics stay the same, you would have actually a reduction because you would have some dilution effect. And then we would want to know, okay, what was going on with vector spread and parasite epidemiology, etc. So now I'll move to a parasite. These are some parasite data from Ribachi, so on the Carolian Lagoon. And this data was specifically collected from juvenile blue tits. And what we found is that from 2003, 2004 to 2018 to 2019, there was a significant increase in the amount of Hemosporidian parasites on the Carolian Lagoon within blue tits. So this somehow supports our first hypothesis, is that if vectors are increasing and the diversity and abundance of these are increasing, then we may see an increased prevalence and diversity of parasites. If we dive a little bit deeper into the specific parasite genera, so we have Hebron proteus, leukocytozoon, and Plasmodium, we see that between 2003, 2004, and the other period, 2018, 2019, we saw a really big jump in Hemosporidian prevalence, but also Plasmodium showed up, which begs the question that, okay, now Plasmodium is there, then there must be suitable vectors that also carry these parasites. And there was a paper by Valkiūnas et al. In 2022 who found similar patterns, although they said that Hemoproteus prevalence remained stable and Plasmodium increased. And here we see that Hemoproteus actually increased. But I think this is where I will stop because I will put a fat warning on this. This is a start endpoint analysis. So I'm taking data from 2003, 2004, and I'm comparing it to 2018 and 2019. And this is generally not what we try and do in temporal analyses because we miss all these fluctuations in between. So I give a warning here because it is not necessarily the correct way to do it. Yes, you can find interesting results, but a more appropriate way would be to have data from this time, this entire period, but this was not there. So lastly, I would like to talk about vectors and specifically how they've changed in Lithuania through time. And I try to also identify what environmental parameters may have impacted the change in distribution and or abundance of vectors through time. So I use various impacts here, but I'll just go into it quite quickly. And if you're afraid of mathematics, look away now, because we have some complicated function here. So this is basically just a hurdle model, which uses some various distributions, but basically broken down. With this mathematics, I wanted to ask two questions. What factors influence the presence of a vector at a site? And two, when these vectors are present, what factors influence their abundance at the site? So this all math just translates to that. So I'm almost done. So if you wanted to be bitten by a vector, where would you go and where should you go? And what this model predicts is that around southeastern Lithuania, this is really a hotspot for vectors. If you want to be bitten by a vector, probably, or if you want to be bitten by a vector, this is where you should go. And one of the biggest drivers here was water quality, which is a bit counterintuitive because you think that mosquitoes can breed in any water, but in fact, it's not like this, and that better water quality means better vectors. And mosquitoes, ceropatogonids, and simulids, they all have different biologies. But if you wanted to get bitten by lots of vectors, where should you go? And again, northeast Lithuania is your place to go. So around Drukšė, we were sampling this season, and we weren't bitten by vectors, but we were bitten by really a shitload of tabanids. So if you want to get bitten by some horseflies, I would go to this area too. And what's important here is that we see a bit of a shift. So I like to think of Lithuania split between the this part and the lower part so is northeastern and southeastern. And here in the northeastern section, we see that natural areas as well as precipitation were major drivers of vector abundance within these areas. But in the southwest regions, agriculture and temperature were the major drivers. So anyways, to sum up, if you want to get bitten by vectors, I please ask you to go to these areas and tell me how many vectors bit you and which vectors bit you, because these models which I made are not hypothetical, they're based on data, but they need validation. So I would suggest that people working on vectors and parasites go to these regions and try and validate this model because it's quite nice. I need to check the predictive power. I also want to repeat this analysis separately for the different vector genera, because each of these genera have their own biologies and life histories. And also I want to look at how these things have changed through time. And lastly, I would say that to the parasitologists out there, go potentially to these areas that are predicted by my model and just see, maybe you find something else that you don't think you would find. So these are some outputs from my postdoc: 4 papers, 2 internships, a conference, 3 invited lectures. We also submitted a LMT group project, which was successful and that we will be working on now. And of course, many memories, a lot of knowledge and many people along the way. So these are some of these people. And thanks to these organizations for helping me bring this to life. Thank you.
Thank you, Nathan, for your very, very interesting and emotional presentation. I expect many questions, but we are short in time, and maybe only one question, and more questions during coffee.
That's more or less the same question as I addressed to previous speaker, talking about such phenomena, limitation within political borders. In my mind, is challenging. And even now, observing your maps, I can identify it, for example, basins of the rivers, which probably have influenced your results. So I understand from a political point of view, title of the project, Lithuania, blah blah blah, it's accepted, but from scientific point of view, I'm not very, very happy about such limitations. Probably that for the next postdoc, project, just enlarge based on probably some natural, phenomena, not political.
The challenge is data availability, and also it's hard enough trying to decipher what some Lithuanian names are. And now I should try and do Latvian and Polish too. It's difficult if I expand to bigger regions. But also geopolitical tensions also stop us from expanding our research. For instance, Russia and Belarus, we don't have data from these places.
But Baltic countries, Poland, it's open.
Sure, but if we talk about the Nemuna specifically, we would need data from Belarus, and this is not really possible at the moment. So, sure, I agree with you that political borders are just man-made constructions, but also there's a void of data from the eastern side of the east and northeastern side of Europe. So more data is necessary, but I consider myself a biological data scientist, and I always need more data, but it's difficult to get it. But I will try and expand.
Okay, we can prolong during discussion.
Good.
Hello, everybody. I'm Sergei Shimchuk from Center for Physical Science and Technology, and I just started the postdoctoral fellowship in your institution. So my supervisor is Vaida Šeirienė, and I want to present some results of this half year that we have already working on. And so I want to present the work named Synthesis of Biodegradable Nanosorbents and Study of their Absorption Potential in Different Environmental Media. So the aim of this short period was to examine sorption potential of eco-friendly nanocomposites for radionuclides and heavy metals and investigate the efficiency in different natural butters. So, but were tasks to synthesize, to characterize the sorbents and make a batch sorption experiments and also ecotoxicology experiments. So what it is magnetite? It's ferrum oxide that is a special structure, octahedral, and it has the magnetic properties. So it's like the magnets and can go to it. So it is easy to separate it from different media, and also it can be also used like a sorbent as alone. And another thing which is much more interesting is the hytosan, which is prepared from histine by distillation, and it is very useful in sorption and different water treatment or something like that. And it is easily interact with as organic metals or inorganic materials or substances, and also it's biodegradable and so on. So what we decided to do is to make the one composite of magnetic hytosan, so it can be easily separate from the media which we are examining or just for treatment of water in different media. So how the magnetic synthesis of the magnetite was done. We we made solutions of ferrum 2 and ferrum 3 ions in 1:2 ratio and put it to the one flask and mixed it for 30 minutes and then added drop by drop ammonium solution, and then it was also heated a little bit, and then we got the magnetite. So we just separated with magnet, purified and dried for better dosage in our experiments, and to wait, as you know, you need to know the concentrations when you're doing something with sorption. So, and then we prepared the magnetic hytosan, and the scheme is, you see, there were two methods. It's cross-linking both, but in one of them I used cross-linking agent, it's glutaraldehyde, and another one just adding two substances, solutions of magnetite and hytosan, and mixing them for a long time and trying to make lead the pH about 8 to ensure the cross-linking reaction. After that, also we separate it with magnet, purificate it, and dry it. So then we characterize it with FTIR. It's infrared absorption, and as we can see in the spectrum, the A, it's blue one, it's a spectrum of magnetite, and the specific area is at peak at 539, and B, it is hytosan, and C and D is a magnetic hytosan 1 and magnetic hytosan 2, which are synthesized by different ways. And we see that in those spectra also that peak appears. That means that the reaction was done and it was okay. It's magnetic hytosan. Another method was used technically. It's Mossbauer's spectra of magnetite nanoparticles and magnetic hytosan, both of them. So we also recorded the Mossbauer spectra, and as we can see, they are mainly showing the magnetite. So in all cases, we can see it and find there is no other different oxides of ferrum, like it can be maghemite or something else. And also we measured the magnetization, and we see that the sorbent that was synthesized by second method showed a better magnetization because it's green dots showing it's higher than the first one, which is blue. Also recorded this picture, and we see that also magnitude was connected to the Haitozan. Dots are the magnitude. Specific peaks, like just to be much more clear to see where they are. And the red one is magnetic Haitozan. As we see, comparing with black one line, it is changed because black one is Haitozan, and we see the appearance of new one, a lot of new peaks that indicates that magnitudes have been connected to the Haitozan, and that's why we prepared the magnetic Haitozan. So also we took the same photos of this magnetic Haitozan, and we see first one is magnitude just, it's dried, so it's like a little bit not as small as it were described during the Mössbauer. Mössbauer showed like the size of it. It is about 17, 18 nanometers of each of crystal of magnitude. The Haitozan to compare, and at the lower two there are magnetic Haitozan, different and we see that the formation of some small particles is clearly seen in the left one photo, and when we zoom it, we see it's covered with the magnitude. So then we made sorption experiments that we used the chromium, nickel, and cuprum for stock solutions, and as target ions, we used distillated water, and in lab we prepared like solution that are similar to the usually find water in the environment, and it is different characteristics like pH 6.5 and so on. The temperature where we were working 20 degrees, and contact time was 1 day, and as we see, we made a solution, diluted it to required concentrations, and then added adsorbent dosage 1 gram per liter, and made absorption tests, then separated using magnet the Haitozan, magnetic Haitozan from the solution, and then measured the concentrations, what is left in the our samples. So here are results of heavy metal sorption, and we can see that sorption capacity was quite high. The blue one is for cuprum, and for chromium and nickel, it was quite normal, like other researchers find, but for cuprum it was better. And sorption removal efficiency was reached about, at low concentration, it was very high, about 98, 99%. And at higher concentration, initial concentration, I mean, they were lower as expected. But the area which is mostly important for us for next studies is the lowest concentration, because they are not far away from the maximum permitted concentration of such metals. So also this data was analyzed using absorption isotherms. So we fitted 3 of them, like Moore, Fredlich, and Hill, and we can see the table, which is with some results, and we can see that all were fitted quite well. The most, the Q max values, it's the max capacity that is calculated according to the isotherms, can be the highest was to cuprum, and others you see not bad. It should be. And then we also made absorption experiments in natural waters, and we used the Baltic seawater, Balsis Lake, and Neris River. Also, we added some amount of stock of target ions and made solution, also diluted it to different concentrations which were required, and added some traces like cobalt and europium to look how it will be if we'll also have such pollutants. And another thing that we measured, after all, the concentrations using the gamma spectrometer. And before, the heavy metals were measured by just mass spectrometer. And one of the most important maybe things that using the tracers, you can work with environmental media, just mark one of the metal that you want to study, and it won't be like difficult to detect after the sorption. So the sorption all process was the same, and after all, it was separated by magnet and measured. So the result for this sorption is on the screen, and as we see, the best sorption capacity was for europium at Belsis Lake, and at all, europium was much more better results received than for cobalt. So it can be because it's much more active, maybe metal. So that's why. And also it depends on the media where it is done, or where experiment was. So also we fitted the absorption isotherms for the data, and we found like it was Lemur and Froelich isotherms, the linear forms and nonlinear. And as we see here, we fitted the nonlinear forms, and also results as we see in the table, we can calculate the maximum capacity for each of metal in different media. So according to the table, we can see the best sorption potential of the magnetic hydrogen is for europium and Belsis Lake. It can be probably because of less solidity than others, but it should be more maybe studied in future. And the sorption efficiency, also the efficiency, as we see, is a little bit lower than in just laboratory research with heavy metals. So it is for europium, it was a little bit higher than 75%, as we see blue one scale, and for cobalt it was lower than between 30 and 25 and 40%. So it's also not a bad result. It means that there were many of different other concreting ions and so on. So here are some conclusions of this study for now. And also in future, we will try to, we are already making the experiments of toxicology on Dania area. So this, everything which was here before done, it's just to calculate the absorption capacity values, and to that we could further to use them in ecotoxicology studies and to predict our sorption. So thank you.
Thank you very much. Questions, please.
Thanks for a nice presentation. I'm just curious, what's the difference between a gamma spectrophotometer and a mass spectrophotometer? Is the gamma more sensitive?
No, it's just gamma. We just collect data from radioactive materials, so isotopes, and with the mass spectrometer we can whole range. But when we use our carbon, we use environmental media, there are a lot of different. So for that purpose, it will be difficult to find, for example, cobalt with this very low concentration. So for that purpose, the market is tracer. You just can measure with the sample and measure just one isotope, like cobalt or rhodium, can be radium, can be enriched plutonium, and so on.
But you don't have to digest the samples beforehand. You don't have to digest the samples in strong acids beforehand, or you
No, you can just add some amount of traces, like in a marker, and the marker you just measure this.
Wow. Okay, thank you.
No more questions. Thank you. And now the last presentation, study of the protective potential of probiotics in combination with nanoparticles, preliminary data. Dalia Matvienko, Supervisor, Nijolė Kazlauskienė.
Hello, everybody. I am Natalia Matvienko. I am head of the Ichtiopathology Laboratory, Institute of Fisheries, Ukraine. Due to the war, I moved to Lithuania and now work on the Natural Research Center, Ecotoxicologijos Laboratoria. I am Ukrainian scientist, which grant from is MSCI for Ukraine Consortium. This program funded by Horizon Europa and the European Union, Maria Skłodowska-Curie project, support research in 21 countries. I researched 24 months and funded by, for my project. My project called Study of the Protective Potential of probiotics in combination with nanoparticle during long-term exposure, juvenile fish assess possible risk. This project study an effect of nanoparticle and probiotic and juvenile fish. It focused on salmon and the tri-stage fish are used as a part on aquatic food chain. The research take place on the Ecotoxicology Laboratory in Lithuania and the Institute of Fisheries in Ukraine. The MCSI for Ukraine program help Ukrainian researchers reintegrated. It gives them funding and support to continue their work when they return home. This project will add the growth on Ukraine university and research. It will also encourage collaboration with the global research community. The study in relevant and global goals in European document, it launched with the Horizon Europe Research and Innovation Framework Program. This program supports research in food, natural research, aquaculture, and environment. Our work also connects with the European Green Idea, which aim to solve environment problems that focus on fish for restoring natural population, conserving biodiversity, and for security. International research cooperation in key to solving global challenges like climate change. This project allows access to resources and best practice Europe. It also addresses urgent issues in Ukraine related to ecology and food security. Nanoparticles as new type environmental pollutant. Their use in nanotechnology have reason content about their impact on ecosystem, aquatic life, and human health. Nanoparticles have unique properties and many use, but their became natural and not well understood. In the context of global climate change, and this presented global scheme that project. Next slide presented freshwater body of Ukraine. It's a very big system. Next slide presented information for water resource. It's problem for war in Ukraine. This presented aim the project. It's the moment in very important problem war, problem restoring aqua system Ukraine. Stage of research. First stage to study a long-term effect of nanoparticle probiotics and complex nanoparticle and probiotics on the health of fish, the phase stage. Take into account factors such as temperature and pH. The experiment was conducted jointly with Lithuanian colleagues. The results were published on the journal Q1. Next stage. It's evaluate how nanoparticle, probiotic and nanoparticle on fried effect digestive system and intestinal microbiota fish. The research was carried out of Ukraine together with colleagues from the Institute of Microbiology and Virology. The result on the study are being processed. Evaluate the protective role of probiotic against nanoparticle toxicity in juvenile salmons. Create a model demonstrating to effect on nanoparticle and probiotic on fish. On the first stage of our research, with colleagues from the Institute of Microbiology and Virology from the Department of Antibiotic, the select a probiotic strain when would meet on requirement could be asked for fish. This slide presented information characteristic for the strain isolate bacillus. A study on effect probiotic based on the strain bacillus of culture of pathogen isolated from juvenile salmonid, and the laboratory presented this slide. This slide presents information for the activity, the strain and culture pathogen fish, pathogen salmonid is presented our work laboratory testing. Next slide presented information histological study, in particular how change in kidneys, liver, and gastrointestinal tract. The next slide demonstration study of microbiota isolated from the internal organ of fish in the experimental and control group. This presented control group and presented experimental group. We can see that there is difference. This next slide presented study of biochemical changes under an influence in the probiotic preparation in fish liver. Presented this slide all to our work, our research. It's our result that all data presented preliminary. I would like to focus our attention. This year, we have conducted a series of experiments of the combination of heavy metal, copper and zinc nanoparticle with a probiotic strain. This result have not yet been analyzed. During my internship, I participated in international conference and seminar on this. This next slide presented publication Q1. Next slide presented publication, main publication and conference, our work, our research. He also actively worked with graduate, undergraduate, and master student in Ukraine during the second period. Summary: study of change fish organism aquatic environment where there are potential toxicant and zinc nanoparticle are of particular interest today to study of their integration with probiotic if of particular interest. Conducted research in different countries of a proposed work plan will allow for comprehensive assessment on the situation and will be useful for the development of research and cooperation between countries. This aspect fish as a model is used not only for natural water bodies with them of restoring biodiversity, but also for aquaculture with them of ensuring food security. The mechanism on impact on fish organism and the potential risk of nanoparticle for the sustainability of aquatic ecosystem under chronic exposure will be carried and mechanism for minimization the toxic effect on nanoparticle using select probiotic will be proposed. The proposed project will be indently the organism and biomarket most sensitive to long-term exposure to nanoparticle will provide and stakeholder will propose it for the creation as database for environmental risk assessment. Thank you very much for your attention. Sorry for my bad English. I study 1 hour. 1 hour.
Thank you for your presentation. Questions, please.
Okay.
How long are you planning to stay here, and how many nanoparticles or types of nanoparticles are you planning to test during that time? Sorry, how many nanoparticles will you test? 10 different ones or 100 ones? Or...
Test nanoparticle.
Yeah, different one.
Yes, of course. We have test nanoparticle. It's not my project. It's work my laboratory. Colleague Semchuk in colleagues laboratory ecotoxicology. My work it's complex nanoparticle and probiotic. I'm studying probiotic effects for fish, not nanoparticle. Yes. Complex for probiotics, nanoparticle, and in my work it's probiotic for protection. It's a problem nanoparticle for fish.
I want to ask you also, did you immobilize your nanoparticles and probiotic bacteria?
It's complex. We prepare complex, no immobilization. No immobilization. It's complex 4 feet. 4 feet fish, juvenile fish, study effect that complex, not immobilization.
Thank you.
Thank you very much.
I think it now we can to close our conference. And before inviting you to the coffee, I want to invite our Deputy Director of Scientific Affairs to the floor and to take some words to our participants.
Thank you very much, because we had very interesting and long discussion, so finalizing was just few. Thank you very much, colleagues, everybody, both postdocs and supervisors. Supervisors mostly, especially because ideas and this process, postdoctoral study process, has became very successful in our institution, I would say. And even today, we can subdivide our participants into groups: those who are finalizing their studies and just people who are more or less in the middle. But in both cases, you have presented very interesting and I would say broad-scale data, broad-scale both from scientific issues, geographical pattern, and ideas you've been working with. So that's great. And there is one more point I would like to emphasize, talking about our postdoc group. It's the most internationalized, sorry for pronunciation, group which are represented nearly foreigners exceptionally. Very few students from Lithuania. For me, as a Lithuanian person, it's sad, but for institution, it's very good. And thank you for promoting idea, knowledge that our center brought. I wish a great success to everybody. And we would like to have you with your next postdoc, projects here or as employees here. So, and I think today our auditorium is a little bit bigger than the last time. And the next one, next year will be even bigger. I think we will find tools for that. Okay, now we have time for coffee. Thank you. Thank you very much.